This is a very special episode of the Stem Cell Podcast, Overcoming Obstacles in Stem Cell Research. Hey everyone, we are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. Today, we're bringing you our first ever live recording of the Stem Cell Podcast, which we hosted at the New York Stem Cell Foundation earlier this month. It's a very special episode where we had the privilege of speaking to NYSCF industry leaders to gain insights into the obstacles facing the stem cell research community and some innovative solutions to overcome them. It was a great event. So let's jump right into it. Thank you so much, guys, for joining us. And also thanks to STEMCELL, the team at STEMCELL Technologies, also the team at the NYSCF, who've been very generous in making the space for us and organizing this. It's an experiment, it's a bit of an experiment doing this for the first time. And I mean, you guys know, when you do an experiment in the lab, you got a lot of people to sign off on it. You got your Embryonic Stem Cell Research Oversight Committee, you got your IRB, protection of human subjects, you got your even environmental safety, everything. You got to get sign off. Maybe for better, maybe for worse, there is no podcast oversight committee. So this experiment has not been vetted. We're in uncharted water here, but I think we're going to have a good time, and we really appreciate you guys coming out to join us. Again, I want to introduce Arun Sharma, who took the red eye here, so thanks to him for making the trip.
I'm good to go. I got my caffeine.
I came from right across town, so it's been easy for me. How was the trip for you, Arun? You made it?
Trip is good. I'm here in person, physically, mentally, spiritually. We'll find out. But no, it's been great. This is my first time here at NYSCF, and just to see some of the facilities that are here, it's just mind blowing to me. I was talking to Dan about some of the automation facilities that they have on site, and initially I just didn't believe him. But being up there and seeing what's in store and what you guys are doing, it's really inspiring. So can't wait to talk more.
All right. Joining us for this experiment, we have Raeka Aiyar, who's the VP of Scientific and DEIB Outreach. Thank you, Raeka, for joining us. Also, we have Dr. Laura Andres Martin, who is a Senior Research Investigator and leader of the NYSCF Women's Reproductive Cancers Initiative. Thank you, Laura. And finally, we have Dr. Daniel Paul, Senior VP of Discovery and Platform Development. Dan, thank you so much for joining us. All right, we're ready to get started. So just to set the stage here, I wanted to tell you about my own experience with the NYSCF. Approaching now 15 plus years ago, I was in the inaugural full class of Druckenmiller Fellows. This was 2006, and it was a very different time for stem cell research. The president was George W. Bush, who presided over sweeping restrictions of federally funded stem cell research. There was a lot of enthusiasm about the therapeutic potential of stem cells, but I think that was balanced, maybe even overbalanced, by a lot of caution, a lot of skepticism, controversy, really, about the necessity for destruction of embryos in order to make patient-specific stem cells. In fact, the whole field, I think, was really fraught. And not only that, but all the things we take for granted now. The idea that stem cells could be reprogrammed to a pluripotent state from terminally differentiated state was like fringe. I think there's a lot of scientists out there, stem cell scientists even, who thought that there was no going back in time, or up the Waddington diagram, so to speak. And I would say more so, there was consensus. The one thing everyone could agree on is that we needed safe places. We needed a safe haven where specifically the younger researchers who didn't have their war chest built over a long scientific career, they didn't have maybe the freedom to explore their ideas because they were living in a world where they had to get a job, right? And they had to get funding for their research. And that's where the NYSCF stepped in. The New York Stem Cell Foundation stepped into this vacuum with the intent of trying to create a safe space. And I can say personally for me, it really was a career-changing, world-changing event in my life, and it really it opened me up to a new world that I still carry with me today. And just with that as a backdrop, I want to start with Laura, just to ask you. You were a NYSCF Druckenmiller Fellow yourself. In fact, you were mentored by NYSCF Druckenmiller Fellow Marco Siendel, who was in the class right after me. And then you yourself became a Druckenmiller Fellow, ultimately now Senior Research Investigator here. So you've really existed in a separate generation maybe from mine, but you've run the span in NYSCF in many positions. Tell us, what do you think is unique about the NYSCF? Tell us about your experience and what sets this institute apart.
Yeah, thank you. Well, first, it's a pleasure to be here. Thank you for the invitation, you guys. And yeah, so I guess my example could be that a little bit of that example of how NYSCF is that family that keeps growing, right? I first heard about NYSCF when I was a postdoc at Marcos in the lab, my first time studying stem cells. So I was coming from a very different background, but always interested about it. And then I remember when I started in the lab, one of the first things Marco asked me, you had to go to the NYSCF conference. Had you heard about it? And you had to apply to the fellowship. We need funding to do this. And then I was like, I don't know what is this, even though I was living in New York at that time for a while already. So I attended the conference that year. That was in 2012. So it's a little bit of generations after you. And then for me, it was just this sense of the innovation, the potential, and how all the community was getting there together, and you just get this vibrant, spirit of how many things you can actually potentially do, and it brings you that spirit into your own work. That was my first exposure, I would say, to what NYSCF community is. And then as I go into this journey, more getting, eventually, I got funded. That was not the first try. It was after some tries. I got funded, and then you get immersed even more into the other. There is the conference community, the big community, but then you get into the family, actually, and how everybody has not only the innovation. I was attending the retreats that we have for the innovator community, the fellows, and just what these people can do. I was amazed. So it inspires you, really. So it really was a changing event for me too, career-wise, but also giving you that spirit or confidence that maybe I can also do things that are amazing. Maybe I can get into that point like everybody else. And then I got here in 2018, working at NYSCF now in our oncology program, women's reproductive cancers, and I think we are getting there into trying to contribute to those amazing things.
Yeah, so NYSCF had humble beginnings, certainly, but fast forward and here we are now. NYSCF is truly a powerhouse in stem cell biology, not just nationally, but internationally. And so let's talk about some of the cutting edge science that's being done here at NYSCF, which is really, like I said, leading the charge when it comes to stem cell biology. And Dan, I alluded to this earlier when I did the introduction. One of the most exciting things I've heard about NYSCF is this automated approaches, and these automated approaches that are being pioneered here. And as an outsider, I didn't believe it. I didn't believe these things were possible. I mean, we hear about a lot of this in stem cell biology. It's like, oh, automated this, automated that. Other folks are striving to get there, but this is the reality here at NYSCF. So, this is the type of potential the type of science that has the potential to make all of our lives easier as cell biologists. But I think there's also some important aspects and important questions of how to democratize some of these technologies, which are so revolutionary and can make our lives so much easier. How do you bring those technologies to the greater community? So Dan, could you talk a little bit about how these cell culture automation technologies are making your daily lives easier here at NYSCF, and also maybe a little bit about how to democratize these technologies?
Yeah, absolutely. Well, just to echo Laura, it's great to have you guys here on site. It's lovely, and to have this audience is great as well. To the skeptical, I've been here at NYSCF for a long time. I started as a postdoc before the robots were even here. And I think even I was maybe a slight skeptic at the time, at the beginning, 10, 12 years ago. but sure enough, over the past decade or so, we've really, I think, built something that's quite unique and really stands alone, especially for stem cell research, but maybe just for biology as a whole as well. So yeah, so we've the platform that we have upstairs really automates all aspects of stem cell biology. So we can go from a tube of blood or a skin biopsy, and we can put that tube of blood in one of our robots, and it will isolate the PBMCs from it. We can then put those into automated reprogramming runs where we don't have to do anything aside from put reagents on the deck or reload tips, maybe a few go into the freezer backwards and forwards. But the robots really take care of everything in between: feeding, passaging, imaging. And there might be, on any given day, a couple of hundred plates in culture. Everything gets fed or passaged during the day and imaged overnight, so it runs sort of 24/7. It's a real operation, but it's a testament also to the people that run it. There's a handful of them in the audience, but as much as we have robots, there's a lot of people that still make this possible. It's not just reprogramming though. We've over the years rewritten things and improved things and iterated upon it to a point where we can do genome editing on the robots. We can do all these differentiations. It's a pretty amazing platform when we stand back and look at it. And I think one of the words that we come back to a lot with something like this is scale. I think that's something that generally in biology, scale is something a lot of people think about, and scale can mean a lot of different things for different people. It can mean lots of cells from any one person, maybe for a clinical therapy. But we think of scale slightly differently. We think of lots of cells in parallel. So we want to be able to take a 96-well plate and have 96 different people represented in each well. And so by doing that, we can get to a level of democratization in a few different ways. One is we can actually have representation in the dish of not just any given disease, but all the different ethnicities that might be represented by that given disease. So instead of just studying 3, 5, 10 cell lines, and I know this is very easy for me to say this with these robots upstairs, but we can put 96 different people in and really start to interrogate the impacts of genetics or different other characteristics of any given individual. I think though, again, where we stand, we're pretty fortunate to have had this, and we gave a little bit of a blueprint when we published this a while back. But we've begun to open source a lot of tools here as well. So we have a fairly big data science or growing data science team, I'll say. And just in the past couple of days, actually, we've released a couple of open source tools based around a lot of our imaging workflows. And so I think it's doing things like that as we can continue to sort of innovate that way and provide things back to the community. We can definitely give some level of democratization in that respect. And then we engage a lot with many others as well. And so it could be that a group might have done an incredible job of collecting biomaterials. Maybe they've collected a large biobank of blood specimens or skin biopsies, but they just don't have the bandwidth or really the desire to sit and reprogram them all. They want to actually be doing the differentiations and the things that will actually lead to maybe an insight into a disease. And so we can use the platform upstairs, whether it's through collaborative grants or whatever the means might be, to onboard those samples here, reprogram them, and then send them back, and allow them to really, use their time and their efforts for doing, real science, if you want to call it that. I think what we do upstairs with reprogramming is real science, but the cutting edge stuff where it's going to represent a disease in a dish, and so democratization probably spans all those different areas, but yeah, I think that touches on it.
Doing real science, but also real good science. So that's important. One thing you alluded to there is, how the platform that you developed, it can incorporate a diversity into the platform itself by, say, representing the greater population of mankind. And of course, we're a very diverse species. One big thing that's being done these days is, of course, focusing on women's health in particular, and representation of proper samples and induced pluripotent stem cell lines in that regard. So we can chat a little bit about the Women's Reproductive Cancer Initiative here at NYSCF, which you're leading, Laura. It's critically important. It's focused on women's reproductive cancers, such as ovarian cancer. And if we think kind of more broadly, this initiative is building on other initiatives at the federal level, right? There are grant and funding agencies that require researchers to address sex differences in their studies. You have female mice included in basic science studies as a requirement these days. And I'm at my own institution at Cedars-Sinai in Los Angeles, all the way across the country. There's a massive clinical initiative focused on women's heart health. So Raeka and Laura, could you talk a little bit about the importance of having and promoting these initiatives focusing on women's health and disease? And what are some of the next steps that we could take for some of these initiatives at NYSCF?
Yeah, I can start for the oncology, in particular ovarian cancer. So that's one of the things that we started this program in 2018. So it's actually a pretty new initiative. And then the problem is that this ovarian cancer has been neglected historically on studying it, funding it. And we realized that innovation was not getting there for this particular cancer type for many different reasons. And innovation is one of the characteristics, I guess, that we can say that we can do here is bringing state-of-the-art technologies, utilizing the latest of stem cells technologies to do this. So in this case, what we do is to have a personalized approach to model the disease. We actually do this by getting tumor samples specifically from each patient. They get surgery, it gets to us. So we can not only growth that patient's specific tumor, but also if you have different tumors with ovarian cancer, that happens in more than 80% of the patients. They are completely metastasized everywhere. You can actually also growth every different tumor from different sites, which is very important for treating diseases that are in that advanced stage. This is based on technology that it was created for growing adult stem cells, actually. So we don't really have to manipulate anything, and that's the beauty of the system. They just do what they are supposed to do in the dish. They recapitulate the tumor completely, molecularly speaking, but also another of the beautiness of this system is that you can put drugs on these tumors. And so far, preliminary evidence for a few drugs is that they can also recapitulate the responses of those patients in the clinic. So now we can take an approach where we not only are creating collections that represents every individual patient and every individual tumor, so that leads to creating genetic associations as well when you have a large collection, large enough, but also you can actually personalize treatments and tailor those treatments to specific tumors. And eventually we would like to get there. So that makes us to be accelerating things in many different ways, because this is focused on women, which there is little focus for many different angles, as you were saying, but also we are advancing like an approach that is cutting edge on the precision oncology space by bringing these technologies as a proof of concept for this cancer type. And we would like to extend that later to other women's cancers in the future.
Yeah, I just want to underscore the tech emphasis at the NYSCF Research Institute is really unique in my view. I mean, maybe all you guys in the room know this from being upstairs, but you listeners at home maybe don't. You go upstairs here and it's, what a science lab looks like, a stem cell lab for the most part, but you walk into the corners here and there of the labs upstairs and you see like computers broken down to the bits. You see all these elements that I never saw in a traditional research lab. And I think what jumps out at me is that we got a bunch of tinkers here in the building, and also this really ground up approach, right? Where they're building the things, and then they got to build the solutions to the thing they're building in this constant recursive iterative fashion over the course of over a decade to arrive at this point with the machine. So just to underscore, it's a different approach where you got to get all these different type of experts in here that I think is really a model to look toward. And at the end of the day, the upshot is, right, it's therapies for all, right? We want to be able to treat all the diversity of human beings and all their diseases. But also, Raeka, here at the NYSCF, there's another point of emphasis. It's not just therapies for all, but it's science, doing science for all scientists and all types. And I think that type of inclusive approach, it has to be very deliberate. We live in these insulated silos where, for the most part, we're looking across the bench at people that are very much like us. And I think that's something that really needs to change in our field, every field, but really in fields that are focused on treating people, right? Because there's all these types of people. Raeka, speak to that. How do you guys, what's the philosophy in terms of DEIB, and how do you make it happen? Not just here at the Institute, but how do you extend that influence beyond the NYSCF?
Well, I'll be the third to echo my gratitude and excitement for this event. And, it's been so long in the planning, so I'm really grateful that you guys are here. These are really great questions. I think diversity, equity, inclusion, and belonging, or DEIB as we shorten it here at NYSCF, is really something that we've considered mission critical from the get-go. And that comes from, our founding CEO, Susan Solomon, who recognized that, we're not going to meet our mission. NYSCF's mission, as a nonprofit, as a patient-focused nonprofit, is to accelerate treatments and cures for the major diseases of our time using stem cell research. And we can't go as fast as we need to go unless we have all of the greatest minds able to contribute to these formidable challenges before us. And, we like to think that science is a meritocracy, but that is certainly not the case because of things like systemic discrimination and the biases that we all sort of carry around. So, we embed that into our approach in a lot of ways. We've talked about, NYSCF having this kind of unique role in the ecosystem. That what we like to have is really just as an accelerator and to kind of identify these unmet needs that we can address. So, the Women's Reproductive Cancers Initiative is an excellent example of that. Ovarian cancer receives about 20 times less funding than, equivalent male reproductive cancers when you adjust for mortality and incidence. Huge unmet need. And that's why we don't see the benefits of precision medicine being realized in diseases like ovarian cancer. We also, we were talking about health equity, and most of what we understand about biology and disease and therapies comes from studies of white European males. So our understanding of what disease is, or even what health is incredibly skewed and incredibly biased. And so there are, we're not serving all of the patients who need treatments. And that's, again, part of our mission as an organization. So this is one major area that we take it on, is in the research lens, is, how can we advance inclusive biological research. And one aspect of this is really, an intense focus right now on building out the ethnic diversity of the biobank of stem cell lines that we've created as a community resource, again, sort of to accelerate progress throughout the community, and through initiatives like the Women's Reproductive Cancers, and through the research that we do in our own labs here to say, like, how can we include more ethnically and ancestrally diverse cell lines that have been so systematically marginalized out of participation. And there's, that's very complex reasons as to why that's the case. But one way that we think we can make an impact is just by, having those lines represented so that the discoveries we make pertain to the populations that need them. In terms of the workforce, it's of course important that, all of these great minds out there who have merit, who have brilliance, are able to contribute to making the field move forward. And so we focus on that in a few different ways. One is through our events and communication. So things like this, really just raising awareness about the issues that, the bias is very well alive here, and all of us have privileges that we are blinded to. I'm a woman of color, so I can see what happens to women of color, but I am also a white passing, hetero, native English speaker. So I get a lot of benefits from that make it hard for me to see what my colleagues not in those categories have to endure every day. And when people are not able to bring their authentic selves to work every day and to feel included, they're going to be that much less productive. And so even if you just look at it from a completely pragmatic point of view, and in addition to it being the right thing to do, we need to change the way that we're doing this. We have a lot of things that we need to dismantle. And so one of the ways we take that on as well is through our grant making, through the NYSCF Druckenmiller Fellowships, through the NYSCF Robertson Investigator Awards. We've been really intentional about changing the way that we do the selection process to elevate minoritized scientists who are not getting as much of a chance and who have overcome undue hardships to get where they are. And to reward those scientists who are fighting this fight, who are going out of their way to be advocates and to create a better world for all of us, because that is the community of the future, and that's what we're really trying to build here.
So let's talk actually a little bit more about that. I mean, I think the timing is pretty good. You mentioned how NYSCF supports internal and external investigators through the Druckenmiller Fellowship and also the Robertson Fellowship Program. I mean, it's no secret that, I'm a junior investigator. It's tough to be a junior investigator these days because, funding pay lines are not so great. You always stress about getting your first major research grant, federal grant. And we've all seen that graphic of how your first R1 these days is delayed into your 40s, and I'm just freaking out right now. But I think NYSCF has, as you alluded to, you've definitely done your part to aid junior investigators, not just internally but externally. In addition to these fellowships, which are so prominent, what else are you doing to support that next generation and cultivate that next generation of scientists? And I think we can open this up to everybody. So Dan, I don't know if you want to start.
Yeah. I think in a few different ways. I think, as you guys have very kindly said, we have a pretty unique resource upstairs. It's surrounded by PIs that run pretty amazing labs here. But for an undergraduate, someone that's just left their undergraduate degree to come in and train in an environment like this, I think is pretty exciting. They get exposed to, maybe they have to go and work with our software developers, or maybe they have to go and work with our engineers or our data scientists. And so at that sort of, freshly coming out of school, to have that instant exposure to those kinds of things then gives them a whole new grounding for making them think about the career trajectory they want to take. Maybe they came in here thinking, I want to be a biologist, and all of a sudden they have a lot of fun working with our product management team or our software developers, and they're like, actually, maybe I want to go and do that. And so I think we're helping people think about what they want to do by exposing them to an awful lot of different things. We do a lot where, at a very different level, we go out to different schools. We might bring a school group in here, or we might go and teach a class at an elementary school or a high school, and again begin to expose them to science that maybe they've read about, maybe they've heard about, or maybe they really haven't, and they haven't thought about what a stem cell is and what it might be able to do. And if you can invigorate and sort of inspire people at that age, that's pretty exciting. We've had people come back to us after maybe they came through on a tour one year, and they've come back and said, that was really inspirational, and it made me think about what I want to go and do in college and things like that. And so if we can continue to do those things, I think that will help foster the next generation of scientists. So there's a couple of different ways we do it.
Yeah. I do want to emphasize what Dan was saying about inspiration, right? Because I think that's the key to make people being successful, right? You inspire them, and it's like, I wanna, even if it's I wanna be like you or something like that, right? And I wanna also point out about wanna go back on other events that we do, in the conference, and from my own experience, even going to different conferences and how you start paying attention to the diversity in there, for instance, it's just something that stands out. And you go to an event or a conference and you see speakers that are from diverse backgrounds, women, men, diverse ethnicities that are balanced on that it's just start to run in your head how, I love coming here maybe because of that. And I think exposing to that kind of environment is also creating the mindset for people to start thinking about this should be even more important, and we should all be thinking about this.
Yeah, I think that's right. And with our events, we do we do try to use our role as a convener to showcase diverse talent, to showcase diverse voices, to really help to correct some of the preconceptions that all of us have, even those of us from minoritized groups, of what a scientist looks like, of what success looks like, of what good leadership looks like. That can look like a lot of different ways, and bringing a lot of those perspectives together is one of the things that we take really seriously for our events. In the broader community, I think a lot of the emphasis that we put into DEIB in our selection process helps to build a community that is attentive to these issues, and helps to build a community that really advocates to fight for correcting the course that we've been on. And so we do programming at our annual retreats and at our annual conference, really dedicated to issues around DEIB, to make sure that there is that engagement. Internally at NYSCF, there are also a few different things that we do. We have affinity groups that are dedicated to supporting minoritized scientists from particular identities, and these are really kind of formed and driven by more junior staff to give them a voice in sort of what is it that we can be doing better, because in leadership we don't always know. There's obviously some things that we don't see, and so it's really important that it has to be a kind of top-down and bottom-up sort of approach. We're fortunate that it has been, top-down from the get-go, as I said, with Susan driving so much of this. But it is also really bottom-up, and we like to, just make sure that all voices can be heard as we try to make the most inclusive environment possible.
Yeah, I think you mentioned Susan. I don't know that anybody knows better what it's like to be an outsider, or knew better, sadly, what it was like to be an outsider and to break into something and change the game, so to speak. Susan Solomon, you talk about the top-down leadership. She really invented it, you could say. Media mogul turned scientific force with no formal training, really one of a kind. I was lucky enough to be at her memorial, which was a sad occasion, but also closed the loop for me. I really encourage everyone listening at home and in the room, if you haven't already, to check out that memorial service online. It's posted at the NYSCF website, because it's not only touching, but it really, I think, illustrates in all the tributes to her why she was such a force. And I think it is because she was an outsider, and she brought all these people together. I mean, I recall vividly thinking, wow, there's Elaine Fuchs, the biggest deal in science, a row ahead of me, and there's Martha Stewart, the biggest deal in something else, maybe everything, row behind me. I mean, it's a real confluence of two totally different worlds that maybe she's one uniquely qualified to serve as a bridge between. But I thought more than anything, the tribute from her son, Ben Goldberger, really underscored and put it all together for me about what kind of a person she was. And this is broadcast, a family-friendly show, so I'm going to paraphrase what he says. But essentially, in describing why his mother, what he thought, why his mother was so successful, was her anthem, her motto, which was, I'm going to defer to the producers here, screw them, except instead of screw, the F word. And that's what she'd say, if someone say, you can't do this, screw them. Her say, your son can't do this, Ben would say, screw him, do whatever you want. And I think that maybe, I don't know, you guys can fill this in, maybe that's in the DNA of the NYSCF. I want you guys to talk about that from your respective positions of authority and expertise in the NYSCF Research Institute. How does that anthem manifest in what you're doing? Starting with you, Dan, what is it about the world saying you can't, and then you guys are saying screw 'em?
I and just to preface that, I think one of the other reasons that she was so incredible is that it was very deeply personal. It was, so much of what established NYSCF in the first place was born out of trying to find better treatments. And in her case, it was for her child, and to find better treatments for diabetes. And that's a we talk about that story a lot still today, and try to retain those sorts of values. But you can see we're in this building because of Susan. But you can see it all around you. If we talk about the robots, just because that's so personal to me, the only reason that exists is because after getting grants and things turned down that like did go out to try and seek the funding to build that platform upstairs, a decade ago people thought that was insane, and like you could never do this, you could never bring this kind of scale or technology to something like stem cell biology. And so in that classic way, she said, okay, well, let's find another way, and we'll go out and we'll use philanthropy, and that underpins so much of what we do here. We are incredibly lucky to have an incredible funding base that is philanthropically driven, and it was effectively seed money for building that upstairs that triggered the growth and the development of that platform. And it needs a figurehead like Susan to be able to go out and inspire someone to write that kind of check that can do that kind of work. It then takes her, she inspired a lot of people then to build that technology. That doesn't happen by itself upstairs. It requires someone inspiring you to work through all the times it doesn't work until eventually it does. And as much as no was not an answer. You couldn't say no to Susan because she would turn around and go, well, why not? And and again, that's why so much of what's upstairs exists. She was a true force of nature. She was just incredible. Many of us would not be in these. None of us would be in these seats today, I don't think, if it hadn't been for her.
I think the way that I try to remember her legacy and the way that it played out for me over the last 5 years when I had the privilege to work with her was, she was just so prescient at noticing and identifying obstacles, because she was just so determined to go as fast as she could. And like others have said, that's because of her, it was personal for her. It was a passion project. And so she could really pinpoint what an obstacle was. So she very quickly understood the obstacle of reproducibility and said, we need robots. She very quickly understood the obstacle of people working in silos in stem cell research in the early days and said, we need a community. She very quickly understood the obstacle of inequity and said, well, we need equity initiatives. We need to fix this because I just, we're not moving fast enough. We're just not moving fast enough. And she just had so much tenacity in removing those obstacles. That's the, not taking no for an answer and, possibilize it. Like if it's not possible, possibilize it. And I think that is what, that passion and that drive is something that carries on in so much of what we do at NYSCF, across the, the communications, the events, across, grants, across I'm speaking to things that I work on primarily. It's just, go, give it all you got, like make it happen. And, the last piece I'll say that she really inspired me with personally is just leading with empathy. I had never worked with a female boss before I came to NYSCF, and she taught a whole different sort of leadership style. She'd be like, how are you? Is everything okay? You don't look like you're okay. Are you okay? Have you seen this show? It's really funny. It'll make you laugh. And, I'd never even had a conversation like that with a boss before. And so I think that just speaks to the kind of culture of the family that we've been talking about where, we're also taking care of each other.
Yes. What else to add, right? But I think like she was just the vision, right? She will came and say, we need to do this. And then maybe because she was an outsider, that's the key, because us maybe thinking about all the steps that it's going to take to get there, you will tell yourself, no. And then I can visualize her just saying exactly and putting this face, why not? Why not? I mean, I wish, because that was the expression, and then you start thinking, just what do we need to do, establish the steps. And the other thing is about family driven, because you are the family, and it's you will do this part, take care of this, other people of the team will take care of another thing. or if we need the innovators community, the stem cell community to come help with this problem, we bring whoever is needed. So she will like convey all the best experts in the world or wherever in the family, asking everywhere. And like, you know, you should not have a problem to, even if you don't know someone, doesn't matter. You go to the door, knock, and like, hey, I have this problem. I heard about you, what you do. Can we work together to make this happen, right? So I think all that like translates into you thinking, okay, well, let's try, because actually you don't try. That was not an option. And then, you know, for the Women's Reproductive Cancers Initiative, actually, it was that personal to her, obviously. She was diagnosed with ovarian cancer in 2017. And I remember her tenacity when she announced this. Actually, it was the year before I joined NYSCF as a scientist at NYSCF. We were on the Innovators Retreat. She stand in the podium. You could see clearly she was undergoing a treatment, but she was, I'm good, I'll fight this. And she was literally there, like her diagnosis was only a few months back, and you could see her with the same spirit, right? And then that's when facing her own disease, and she realized, are you treating this with chemotherapy still? Like still there is nothing else that works? So she educated herself to the point where she could like give a lecture, not only on ovarian cancer, by the way. She will like give a lecture on stem cells technologies like anybody, any of us, really, even if her background, as we said in the beginning, was an outsider, a lawyer, which I learned this, again, going back to my story in 2012, I Googled who she was, because I'm like, who is this person? And Sarah, and it's a lawyer. I was like, interesting, but I mean, and then I faced it over the years what NYSCF is, or the stem cell community that was built is, and I'm like, how is this possible, right? But maybe that's the trick, right? To bring different minds from different backgrounds, expertise. The array is an example as well. There is scientists, there is engineers, there is artificial intelligence people now, and we all together face problems differently because your background maybe give you a different mindset to face those problems, right? So that community really bringing it together made things to happen. And to close up on this again, back to her personal story with ovarian cancer and how she literally had to face it that personal and say, no, I don't want this to be like this for the next few years. And if it's not for me, for the rest of women diagnosed with this cancer, we need to do something. So what's the latest? And that's when I came in here and we just went to the latest stem cell-based technology, and we built, or we are building this platform to try to accelerate novel treatments, because that's what really we need for this disease. Hoping that one day we will do that, and this hope had to be a yes, because there is no as an answer. So we will get there.
Yeah, we've been talking about how personal it was, or the journey began for her from a very personal place with her son and diabetes, and then her own struggle with ovarian cancer. But then kind of it expanded, right? I mean, you guys all became her family. It was her immediate family, then it was her NYSCF family, and now it's like all of us, the humankind, in terms of the scope of her interest in trying to address disease. And you guys are pushing it along here at the NYSCF Research Institute. The end game, obviously, is to translate these therapies, right? But that's like a huge lift. The resources you need. NYSCF is nimble and you know, really effective in their approach, but you need such a different scale of resources to take this into humans in the multiple stages of trials, et cetera. Where are you with that, and what's the horizon look like?
Yeah, I'll take it first. So yeah, so how do we go from a stem cell model of a disease to having maybe a therapy that is actually going to treat, prevent, cure a given disease. We've touched on it a couple of times. I think we're going to do it in a few different ways. One of them is that we need to bring, as Laura sort of said, lots of different backgrounds together, and those are not just of people's different personal backgrounds, but technical backgrounds. And so one of the things we do here, as Laura said, is bring together these engineers that might be computer engineers, they might be mechanical engineers, biologists coming from genome editing backgrounds, from disease modeling backgrounds, from completely unrelated biology backgrounds. And then combining that as well with data scientists, software developers, you name it. If you bring people of just different technical abilities together, really incredible things can happen. And so for us on sort of working on the platform upstairs, we're thinking about how do you improve the modeling, the detection of a given disease, so that you can have better readouts, or not better, new readouts, things that are just going to maybe give insights into a disease that we haven't had before. And again, combine that with scale, combine that with us being able to stratify across individuals or different ethnicities. Maybe then as well, we can start, in some of the screening work we're doing, maybe you can start to uncover that a given drug is going to be better for people of this ethnicity, or they carry this risk variant. And that the way you're approaching treating drugs right now, by starting a trial and just recruiting a whole group of patients and hoping that that drug will work, actually maybe we need to rethink about how we're structuring clinical trials, and that if we could better predict that a molecule that we have in development is going to really only target this subset of people, well, maybe we need to recruit that subset of people so that then the trial might work. It's a smaller, target that you're going to hit, but it's a true clinical, then you've met a need. You obviously hope that maybe you're going to uncover better therapeutics that will, work across all individuals. But unless we're testing on all individuals, we're going to, as Raeka sort of said at the beginning, if we're only testing on a couple of different individuals, that's not going to give us the representation of everybody here. So that's sort of one approach from a sort of a screening perspective to some extent. Obviously, the one that I think most people tend to drift toward when they think of stem cells are therapies using stem cells or their derivatives. And so we're seeing an incredible time in therapies reaching the clinic. We've got people in the audience that are really leading the charge on this, that have been able to go into patients in the past couple of years, whether it's for Parkinson's disease, whether it's for AMD, whether it's for I'm struggling to think of another one, but there are many more. And so we've embraced that here as well. I think one of the things that's very challenging in the stem cell therapy space is access to things as simple as clean rooms. To be able to generate cells for a therapy, they have to be generated, I'm sure many of your audience know this, but generated in these ultra clean environments, these GMP qualified facilities, and there just aren't enough of them. It's very challenging to find enough space to be able to move things forward through a phase 1 or a phase 2 trial. And so upstairs, beside our labs, beside the robots, beside everything else, there's a GMP space up there. And so we're embarking on a clinical trial in macular degeneration, working with institutions around the city, so that we can, again, translate all of the work that we've been doing here over the past 15 years and actually then begin to reach the patient as well.
If I could add one thing to that, the other piece of it, aside from all these internal things that we've built, as Dan mentioned, is that, we are here to collaborate. So we've collaborated with, I think, over 100 organizations in the last couple of years. And the nimble, agile sort of approach we take also pertains to our collaborations. So we collaborate with every kind of organization possible, especially like those who are going to help us to translate the ideas that are being developed here. So that also pertains to everyone in the audience and anyone listening. We're very open to that. So please do get in touch if you're interested.
Yeah, I've said it a million times on the podcast. I think we're in the midst of a golden age in biomedical research, and in particular in stem cell biology, which is enabled by many of the technologies that NYSCF is pushing forward and the community is pushing forward. How easy it is to collaborate these days across the world, across the country. And these technologies are ultimately enabling clinical translation, which is the goal of many people in this particular audience. And on that particular topic, I know the NYSCF, has a particular conference very focused on clinical translation and translational stem cell research. Raeka, do you want to talk a little bit more about that?
Yeah, absolutely. So this is going to be the 18th annual conference that we're putting on this October 23 and 24 at the Rockefeller University, just across town here in Manhattan. And this is, we focused on translational stem cell research from the get-go. I think the conference was one of the very first activities NYSCF ever did. And it was really, to continue that work of breaking down the silos, enabling the collaborations, the discourse, the ideas, covering the hottest topics of the day, like, organoids, cell therapies. We'll talk about drug discovery this year, diversifying stem cell biobanks and the challenges surrounding that. So more is on our website. We'd love to have all of you in the audience come. It's nyscf.org/conference, nyscf.org/conference. And our call for abstract is still open. If you register by August 30th, we're actually happy to offer a discount code for those of you in the audience using the code all lowercase STEMCELLPOD, STEMCELLPOD. Yeah, that's a nod to you guys. So. But we hope to see many of you there. It has the advantage that it's kind of a single track sort of a thing. We try not to, we love ISSCR, but it can get a little overwhelming trying to be in 17 rooms at once. I don't know how you guys did your podcast episodes every day there. That was a tour de force, but yeah, ours is, just a little bit smaller. So, hopefully that enables those connections.
Not smaller, more intimate.
Ooh, okay, yes. We'll take that.
But seriously, I will say that one of the great things about the NYSCF is how small the community is, although it's grown much larger since like 100 years ago when I was a Druckenmiller Fellow. But the community remains small and tight-knit. And to this day, I go to the ISSCR and I find I'm hanging out with all the Druckenmiller alums and just, having a laugh. And I think that's the kind of safe space that's been created at the NYSCF, which is really appealing to me and probably many others. So yeah, this has been really terrific. A lot of fun, a bit of an experiment. Hopefully they won't need to initiate a podcast oversight committee after this, hoping it's not too big of a debacle. But in the interest of trying to share some thoughts and get some feedback from the audience, we have some time in the last 10 minutes or so if anybody wants to ask any questions, preferably not of me. Ask some questions of these guys over here. They've got all the answers. Is there anybody?
So I guess the title of this was Overcoming Obstacles in Stem Cell Research. What may be the biggest obstacle for NYSCF specifically?
Just to repeat the question for the audience at home, and the question was, given that the title of this entire audio experience is Overcoming Obstacles, have we not talked enough about the obstacles we're overcoming? He didn't exactly put it that way, but I think that was the question behind the question. So I put it to you guys. What are the biggest obstacles? Feel free. Each one of you guys, would be lovely if each one of you could weigh in your respective expertise.
Okay, great. I could start. I would say the obstacle that I'm probably most focused on that, I think NYSCF is well positioned to address is the inequity in biomedical research, both in the, the patients that we're trying to serve and in the workforce that's contributing to making those discoveries. So that is why we're doing all that we do in that regard. So, yeah.
Well, I guess what I can say is accelerating things, moving things faster by bringing this community together. Actually, again, we go back to this idea of teamwork. 1 + 1 is 2, and I think that realized also for advancing these technologies, because you can brainstorm together, do things faster, and then at NYSCF specifically, I think the fact that we can do these technologies because of, the funding can be restrictive in traditional sources. So philanthropy has allowed actually to move these things faster than it would have been without that. So I think we should also give the importance of what it has in that sense.
Where to begin? There are a number of different obstacles, and it can vary from very minute things. I wish that manufacturers of equipment would make better APIs so we could like hook things in easier. Very niche request. I think in bigger, more practical things, it's a problem for us, but maybe I'm going to turn this a little bit to the field as well. And there's two things. One is accessibility. And so, yes, it's great that we have the robots upstairs. We're very fortunate. It is expensive, and it is challenging at a technical level to do this unless you have the technical expertise. We really need to kind of break those barriers down so that more labs can bring robots in and scale their research. It can't just be a privileged few here and there that have the ability to do that. The other one I think is going to be, again, there are many. One other one is scale, and a different kind of scale than I mentioned earlier. But if we're going to make therapies accessible to everybody, we're going to have to lower the price of making material in the first place. Like these are expensive treatments. They're sometimes bespoke if you look at like the CAR T space, but in the stem cell space, the trials right now require patients to go on immune suppression for the most part. And so how will that, how that bears out, we'll see over the coming years. But if we can get to a point where we're making stem cell therapies that are personal, again, we're going to have to lower the cost and the ability to do that. And even the scale, like even if you want to go to a stage 2 clinical trial, to generate enough material for this is going to be enormously challenging. And so it's going to take, maybe automation. It's going to take technology leaps to be able to get to these points, let alone phase 3 and actually delivering this as like standard products off the shelf. There are some enormous challenges to still overcome in that space.
You're making me kind of bummed out, Dan.
Well, at the same time, let's turn it around then. It is very exciting that we're even here talking about the fact that there are therapies in the first place in trials right now. I think it's a double-edged sword. It is tremendously exciting that we're going to have, that we have dopaminergic neurons being injected into brains that might actually prevent the progression of Parkinson's disease. That is, the fact that we're talking about that is pretty mind-blowing, like from where we've come over the past 10 years. But it is a real problem that we want to make this accessible to as many patients as possible. And, yes, it's a little sad that is the side of it, but we need to be thinking about that now so we can be building the technology now to meet those needs in another 5-10 years time, whatever it might be.
I want to be clear. I was joking. I'm not bummed out at all. I'm excited. And truth, it echoes what Laura was telling me before, which is that she was saying we fail hard, we fail early. And I think that's the key. The fact that you have a long list of grievances, I think, is where we want to be. It's not that you're pessimistic, it's that you've seen it all. You've seen all the stumbling blocks, and sometimes overcome them, sometimes not, but at least we know, all the unknown unknowns, not all, but enough of the unknown unknowns have been elucidated for progress. So I'm excited. I hope we're all excited. Dan's upset, but
I have my grievances.
Clarify that.
Yeah.
Back to the floor. Anybody else? Any questions?
I got a comment there. When you introduced this as an experiment, right? And so, as scientists, I think we all, this room agrees, this is a great experiment, right? As a reviewer, you request, a repeat experiment of this life experience.
It's a good
How many
point.
revisions?
No, it's just we start with repeats.
Yes. Is it reproducible? We'll have to see. You guys can invite us back.
We'd love to have you back.
Thank you.
It'll be our pleasure.
Well, with that, that brings us to the end of our show, this first inaugural in-person live stem cell podcast recording. It was really great. I think this is a successful experiment, in my opinion. It was really interesting to hear about the technology, the community, and what you're doing to address equity in your community as well. And also just from a personal level, as somebody from the faraway land of California who is not as familiar with what NYSCF is doing, it was really touching to hear about Susan Solomon's influence in your lives and her influence on NYSCF and the greater stem cell community. She's certainly missed, absolutely. So thank you so much to NYSCF. Thank you so much to STEMCELL Technologies for supporting this event. Thank you to our panelists, and thank you to our audience for joining us and making this such a wonderful experience. Thank you so much.
All right, you guys, that brings us to the end of our first live recording of the STEMCELL Podcast. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes. You can also reach out to us on Twitter at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. This was such a great event, and we're hoping to do more like it, so stay tuned, and maybe we'll see you next time live and in person.